Hello everyone, my name is Akashdeep Singh and today we'll be talking about predicting passive blood-brain barrier diffusion. So, have you guys ever been denied to ride a roller coaster because you were too short? Well, the same thing happens to drugs that try to pass the blood-brain barrier. So, what's the blood-brain barrier? The blood-brain barrier is a barrier that blocks the pathway to the brain, so it controls what goes in and out, and also it's a barrier in the bloodstream and the extracellular space. So what does it allow? Well, it allows drug-like compounds into the brain. So if we look to the right, we have the brain, and this is the blood-brain barrier structure. We have the astrocytes, which are like the cell wall, and we have the endothelial cell, which is the interior surface space of the blood vessel, and all the compound needs to do is go through this and go to the brain. So you must be thinking, what must be the problem? The problem is that the blood-brain barrier only allows a select amount of drugs to pass through it, since it's trying to protect the brain from toxins and also give the brain nutrients, which is very important. So what's this review all about? So in this review, researchers are trying to find more drugs that'll penetrate the blood-brain barrier. Also, they want to propose a new predict blood-brain barrier permeabilities from clinical phenotypes, and also test out side effects, what these drugs can do when they pass the blood-brain barrier, how can it affect the brain, and all the other side effects that occur after taking a drug. So how can we design the blood-brain barrier permeability drug design? Well, we need to look at Lipsell's 5 rules. And these rules let us know the chemical properties and the physical properties of drugs. And how is this important? It's important because it helps us tell the difference between a non-drug and a drug. So if we look to the right, a drug cannot have more than 5 hydrogen bonds, and it cannot have more than 10 hydrogen acceptors, and also it cannot weigh more than 500, and also the coefficient P cannot be greater than 5. So these type of rules let us tell which is a drug or not, and we only focus on those drugs and not drugs. So we focus on those drugs and test them out to see if they're able to penetrate the blood-brain barrier. And the methods used for the drug discovery. So in this research, they're trying to test out small molecule compounds due to their diffusion, size, charge, and permeability. So if you look to the right, we have all these 12 compounds that are being tested, all these drugs. Some you might have heard of, ibuprofen, Tylenol. All these drugs, if you notice, they're pretty small compounds, but we need to figure out which are going to be able to be accepted by the blood-brain barrier. So in order to do that, we're going to use the molecular dynamic simulation. The molecular dynamic simulation, it's pretty cool because we can look at these drugs at an atomic level. Since these drugs are really small, so it's really helpful. On the right, it shows the data of which drugs will pass the blood-brain barrier or not. Where BB plus means drugs will cross it, so these are the dark blue to green. BB minus compounds means drugs are not going to cross the blood-brain barrier, so these are the top compounds, the dark red to yellow. So let's look in deeply. So on the left, we have all the drugs, and we have this red dotted line. This red dotted line is where drugs will pass it or not. And as you notice, the plus, we're going to pass it, and minus not. So we can take this even more at this graph, where we can look at the distance from the center of the bilayer. And the bilayer is where the diffusion barrier happens. So all these drugs start at the same spot, and as you see, as these graph keeps on going, the distance decreases and they go to the diffusion bilayer. The drugs that will cross it will go to the negative side, and drugs that don't cross it will go to the positive side. So as distance comes in, you'll find out which drugs on the 12 we listed, or the colors if you look, see if it will go up or down. So the results with the molecular dynamic simulation, things we're testing for. We were testing for the concentration of the drug in the brain divided by the concentration in the blood. Also, we were testing the permeability surfaces area product. And also, lastly, the effective permeabilities. So, we have the results on the right with the dynamic simulation, and the left is a key indication. So we have log BB, which is the concentration in the brain divided by the concentration in the blood, log PS, permeability surface area product, and log PF, effective permeabilities, and also plus meant cross and minus means doesn't cross. So these are our 12 compounds like I showed before we tested. And the main things to look at is the plus or minus. So all these minuses are going to correspond with the concentration of the drug, brain divided by the concentration, the blood, the permeability surface area, and also the effective permeabilities. One cool thing about this simulation is that it predicts before it does anything. So it predicted everything. But one thing you will notice is that ibuprofen was predicted to be negative, but instead it became positive. So this prediction is really accurate, like I would say 99%, but that 1% could be wrong, which ibuprofen was. So the update on the research. 2003 there was 181 drugs predicted. 2012 110 100% identified to penetrate the blood-brain barrier. And if you can look to the right, we have the drug categories. We have the central nervous system, which is connected to the brain. So of course this would allow the brain to work better. So the blood-brain barrier would allow this to go in. And nutrition is also important for the brain. So of course these type of drugs were able to penetrate through. And then we have these other ones, but these were the high percentage ones. So these are the most important ones, if you notice by the percentages. And some innovations. Artificial intelligence. It's a really good innovation since it helps us assist to find more drugs, and it's quicker, and humans don't have to waste time, countless hours just looking for drugs. So we can look at the process of the AI. So we have a drug discovery, and then does multiple trials, and then after those multiple cause effect trials, we have the drug approval, and that can be a potential penetration. So one thing cool about the AI is that it uses its personal knowledge and it learns from it, and then it keeps on adding on, which is a really cool feature. Like I said, performs trial and error, which saves more time for humans to look at other stuff than just doing trial and error constantly with all these drugs. And the AI can predict more stuff accurately with less error compared to a human. What's the goal? So the goal is that we're trying to find out more drugs that penetrate in the future. And the future holds great for this research, since in this research there's millions of drugs. So more drugs will be found, just like how in 2017 110. So 2021 180 drugs were found. So it's all about how technology is going to keep on increasing. We're going to have tools like the molecular simulation where it's more advanced and help us find more drugs. Thank you for listening. Any questions, you can let me know.